Hello, and welcome back to the Athero Talk podcast, the series brought to you by the EAS Young Fellows. I'm Florentina Porsch from the Medical University of Vienna, and I will be your host for today's episode. Today we have a special episode featuring one of the 2024 EAS Young Investigator Award winners, Dr. Mate Kish. Dr. Kish received this prestigious award for his groundbreaking research on the role of complement factor H in atherosclerosis, published in Immunity. So in this episode, we will explore a mix of topics. So from Dr. Kish's personal journey into research to the key insights from his award-winning study and what the future holds in this field. So without much further ado, Dr. Kish, welcome to Athero Talk.
Hello, Florentina. First of all, thank you for having me. I'm really excited to be part of this new initiative. I actually listened to the previous episodes and I really enjoyed them. So it's quite an honor to be interviewed after a set of such well-established and inspiring scientists.
Well, thank you. Great to have you. So maybe to get started, could you introduce yourself a bit more to our listeners and tell us some more about your research background?
Sure, yes. So I was trained originally as a medical biotechnologist in my hometown, which is in Pécs, so in Hungary. And basically, this is where I became interested in immunology. And after my studies, I applied to a PhD position in Vienna to work with Christoph Binder. So my doctoral thesis with Christoph was focused on understanding the inflammatory component of atherosclerosis, and in particular to study the complement system. And after my PhD, I moved to the United States to work with Dr. Philip Swirsky, first in Boston at the Massachusetts General Hospital and then at Mount Sinai in New York. And I'm currently back in Vienna as part of the return phase of my postdoctoral fellowship, while still being affiliated with Phil and his institute.
Okay, that sounds great. You've already mentioned it a little bit, but every researcher's journey is, of course, shaped by key mentors and influences. So maybe you can tell us a bit more about who have really been the most significant figures in your career so far.
Yes, first and foremost, I must definitely mention Christoph, who, as I said, was my PhD mentor, and we are currently working together again. So I really owe a lot to him for his constant support. And I think it's not so often, even in, everyday life, that one can meet such a kind of supportive person, let alone in a professional setting. So as a boss and a mentor, he was very supportive towards me and always sought the best for me. And I feel like even in situations where this may not have been the best for him, for his personal interest. So I really cannot thank him enough for that. And I have to say also, I think it's quite remarkable what he does for the EAS community as well, including the Young Fellows program. And actually, it was Christoph's idea back then to suggest to me Phil Swirsky's lab as a target for my semester abroad, which was a kind of an extra that was included in my PhD fellowship. So we contacted Phil, and then the first time I moved to Phil's lab, it was actually back in 2016. And this was probably one of the most influential half a year in my entire life, both in a professional sense, as well as for my character development, moving to a continent that I've never lived before. And also, finding my ways without knowing everybody there. And then Phil's enthusiasm and passion for science is really contagious. So he really kind of inspired me. He thinks outside the box, and he has also always been very supportive of, following up basically every idea that I've had. And at last, maybe I also would like to mention Cameron McAlpine, who I worked together in Phil's lab, first during my exchange years, and then as a postdoc. And we also maintained a close collaborative spirit after he established his own lab in New York. And I have learned a lot from him, not only in an intellectual level, but also about kind of how to be an efficient scientist, time management, planning. So I can also really thank him a lot for that.
Yeah, that's really great. Also very nice that already during your PhD you got the chance to really work in two high-profile labs and also start building your network there very early on. But maybe a slightly more different aspect. So we all know that balancing research, teaching, and other professional commitments can be quite a challenge at times. So how do you manage it all? And do you have any specific hobbies that help you unwind?
Oh, yes, of course. I mean, in this regard, I don't consider myself a very exciting person. Really, what helps me to recharge in my free time is, spending time with my girlfriend, my friends and family. Besides that, I'm a passionate football player. So I go to play football twice a week. And I also go, running or swimming sometimes, an additional once or twice a week. And maybe another hobby that kind of also relates to moving that I really developed a long time ago is like long walks. I love long walks. And I think I really mastered this, while living in New York City. So on a less busy day, I would pick a borough or an area of the city, and then I would just spend the whole day walking. So, and this is something that you can do no matter where you are. So even in Vienna, I do it a lot, sometimes with company, sometimes alone. And then you can always learn and discover something new while your mind can just, wander, think about experiments, for example, or just, talk to someone on the phone. So I think it's really rewarding because especially in cities like New York City and Vienna, you pass by a lot of tourists and you see the excitement and happiness, which can be really contagious and also give you like an uplift. So basically my summary is just I'm trying to sustain my physical and emotional stability, which of course can be, challenging at certain times depending on the workload. But in this regard, I consider myself lucky because even in Vienna or New York, I'm surrounded with people, also in a kind of my personal life as well as in a scientific setting that are really supportive and always ready to give me a helping hand or just, have a conversation with me.
Yeah, that sounds fantastic. And I couldn't agree more, especially walking is really great to reflect on some of your, of course, experiments, as you said, but also to clear your mind. And you've really clearly lived in the right cities for that with Vienna, New York, Boston. Great. Okay, now going maybe a bit back, focusing more on your research. So looking back, is there anything you would have done differently in your research so far? I mean, I know you still have a long, bright career in front of you, but so far?
Yeah, so that's a absolutely fair question. And then, as we just discussed about walking, you often reflect, what like past years or past days. But, I wouldn't say so. I don't think I would have done something differently. I mean, if I were to think a little bit longer, probably I could come up with a couple of examples. But I think it's maybe natural that at certain times you feel like, oh, this I could have done faster, or, I should have pushed an idea a little bit earlier. But looking back into the time that I spent in research or in academia so far, I feel like everything has happened to me at the right time, in the right setting. So I never have the feeling that I should have achieved more or I would have deserved more. So I think I'm in a happy place at the moment.
That's really, really great to hear. Okay, so maybe let's dive a bit more into your award-winning publication now. So how would you explain the main topic of your research to our listeners? And what do you think makes your findings particularly significant or, let's say, unique?
Mm-hmm. Yes. So as I mentioned before, when I went to Christoph's lab, basically we studied, as a PhD student, I studied the inflammatory component of atherosclerosis. And my studies focused on understanding how the complement system is involved. Obviously, if I were to kind of try to explain to you the complement system, which can be, a fairly complicated topic. I don't maybe go into that much details, but I think many of the listeners know that it's an ancient effector system of innate immunity. It basically acts as an amplifying cascade, and it has been initially known as its effect on clearing invading pathogens. However, it has also crucial housekeeping functions in like mediating the non-inflammatory removal of dying cells, which is of course key in atherosclerosis. So while complement has been long thought to be only active in circulation, it is now evident that almost every cell type, including immune cells, can produce these complement proteins. And on top of that, pioneer work that was led by Claudia Kemper's lab has shown that complement activation can even occur intracellularly, where it modulates like basic cellular functions. So this was huge, and it opened a new chapter in complement research, and that was also very interesting to us back then. So as a consequence, in this study or in this paper, we kind of speculated that at sites of chronic non-resolving inflammation, like atherosclerosis, local complement production that can be fueled by infiltrating leukocytes may outweigh the importance of liver-derived circulating complement. So in fact, what we then found upon hypercholesteremia, that monocytes that infiltrate lesions actually upregulate complement component C3, which is the central complement of complement, and it's also its natural canonical regulator, complement factor H, or CFH. And this was the study that this is what we looked at more in details. And what we found that while liver-derived circulating CFH has like very important housekeeping functions in preventing complement overactivation, systemically, for example, in the case of kidney diseases, what we found that in monocytes at sites of inflammation, complement factor H actually restricts important pro-resolving functions that are exerted by macrophages, and one of them is C3, so complement-mediated efferocytosis, and this can contribute later on to the necrosis of the plaques. So when we got rid of CFH, specifically in the hematopoietic compartment, so from monocytes and macrophages, which we could also show that these are the only cells that really produce CFH in the hematopoietic compartment, we saw that C3-dependent efferocytosis was more efficient, and this strategy actually led to decreased atherosclerosis and higher plaque stability. So, sorry if this was a little bit too long, but if I were to summarize, what I just said before, I think the reason why we kind of tend to believe that this study was significant is because this was the first description of a canonical complement regulator controlling cell-autonomous functions in macrophages, cell-autonomous complement functions in macrophages. And this could exemplify the relevance of local complement activation versus systemic complement activation in an inflammatory setting.
Yeah. No, that's very exciting. I mean, it really sounds like a key physiological process that, could play a role in so many other pathologies as well. So really great. So were there any more unexpected discoveries or, big moments of insights during your work?
Yeah, certainly so. Because, this already the finding that we made regarding macrophage-derived CFH was unexpected. So if I can give, the whole story a little bit of a context, when I joined the lab, the lab just published like a big paper on the role of complement factor H in modulating oxidative stress by neutralizing an oxidation-specific epitope, which is malondialdehyde. So the natural next step was to study this in experimental atherosclerosis. And even before I joined, my colleague Nicolina, who is also the co-first author of the study, had already started breeding the mice that we needed for the experiments. And these included also triple knockouts. So as you can imagine, that took quite some time. So we had quite some time to go until we could perform the big study, athero study. So then I basically spent the beginning of my time in the lab with learning new essays, and also I tried to read as much as possible on the topic. And even back then that there had been already quite some evidence regarding the capacity of monocytes and macrophages to produce complement factors, especially complement factor H, but mostly in like cell culture models. And no one seemed to test this in an in vivo setting. So therefore, we collectively, quite early on in my PhD, we decided to do a bone marrow transplantation study to answer whether hematopoietic CFH, has a role in atherosclerosis. And to our surprise, what we found that the lack of CFH was actually atheroprotective. So lesions were smaller, more stable, which was completely on the contrary to our expectations. Mm-hmm. If you would consider CFH's role in like complement regulation or in preventing, damage done by oxidative stress. So therefore, we had to come up with a new hypothesis, and we had come up with quite some along the way, and wait whether the results of the full body knockout studies were going to be affirmative of the findings that we made in the hematopoietic study. So these were truly exciting times, and I obviously have to thank all my colleagues for their enthusiasm in this journey, despite the fact that actually, these key observations back then seemed to be in part counterintuitive to previous work that was generated in the lab.
Yeah, which is, of course, always a challenge, but actually even more exciting.
Absolutely, yes.
Results are unexpected. So maybe sticking a bit with the topic of challenges. So beyond the maybe unexpected results or initially unexpected results, and also the generation of slightly complicated mouse models, which I can fully understand working with mice myself. But what are some of the biggest challenges that you faced during this research, and how did you overcome them?
Yeah, certainly what we just discussed, it was a challenge, but as you said, it's like can be a happy and exciting challenge. But probably one of the biggest challenges that we all faced eventually is that before the completion of the project, I moved to Boston to work with Phil again. A decision, by the way, that was fully supported again by Christoph. So shortly before I actually made the move, first of all, it was COVID, so tough times, but also we got a rejection, from science. And based on the reviewers' responses, it was clear that, we should probably change our narrative, that maybe we have to find new ways to kind of really emphasize the true value of the manuscript. So we had to rewrite the paper, while operating in different time zones. But eventually we submitted the story to Immunity. And then with the revision, there came, the second challenge, that while we had some of the data that were requested by the reviewers at hand, there was still quite some experiments, revision experiments that I could not do myself physically because I was not in Vienna. So, but however, thanks to the enormous help of my colleagues, first of all, Nicolina, but also actually to you, Florentina.
He's very happy to help.
Yes. So thanks again. And we could really perform all necessary revisions in a reasonable time. And in the end, to our, great happiness, we got a favorable response.
Yeah, great. And it's also always very nice when you actually get helpful comments from reviewers. Of course, rejections are a bit sad, but also...
It was a very kind of happy rejection. Really, the reviewers' comments were in general very supportive.
Yeah, great.
And then eventually we could also address them. So in the end, it's a happy end.
Absolutely. So... were there any limitations during your project, other than, of course, COVID times and the challenges you've mentioned? But
do you think of experimental limitations or more like? Yeah. They're always. I really like actually. So Immunity, or I think CellPress in general, has this new kind of initiative to have like a limitation of the study section. So that I think is really helpful. Yeah. And then even for this paper, we wrote one. So definitely there are some limitations. One that I could think of, which kind of really bothered me, during the whole process or throughout the completion of the study, that the flocked CFH mice were not available. Yeah. So we did not have the means to create them. So we used several complementary methods to show that it's only monocytes and macrophages, but we never really used the cell-specific knockout model, which could really seal the deal. And also, as I mentioned before, intracellular complement, which is like a fairly new field. And we made attempts to kind of try to answer whether the effects that we see with regards to the regulatory capacity of CFH to control C3, whether this happens intracellularly or it's more like an autocrine effect. But we basically never could really show this, or we eventually stopped at this point, and this is, still need to be shown in a follow-up manuscript. And one other thing I could think of is we had a very fruitful collaboration with the University of Würzburg, in particular with Clément Cochin and Alma Zernecke. And then with the help of them, we could show that there's a distinct subset of inflammatory macrophages in human atherosclerotic plaques that express very high amount of C3 and also co-express CFH. However, we have not tested, the functionality aspect of that in a human setting. So this is still something that could be done and hopefully will be done in the future.
Yeah, that's really great. Sounds like there's a lot of follow-up projects stemming from your paper. So very exciting work ahead, hopefully. So one aspect that's, of course, always interesting to all of us, but maybe particularly our more junior listeners. So do you have, or did you have any specific research funding for this project?
Yeah, so for this particular project, which was in my PhD project, so the funding was provided by the Austrian Science Fund. There I was a PhD student enrolled in a doctoral program, which was called CCHD. So that stands for Cell Communication in Health and Disease. So, and this gave basically Christoph and I 4 years of funding. It was an interdisciplinary program that really brought together immunologists, vascular biologists, neurobiologists, together. And it was really, we got a broad and diverse introduction. So we, spent some time in each other's lab. We got familiar with different disciplines. So, and also, as I mentioned before, basically this funding also allowed me to go abroad. So as I meant, into Fields Lab back in 2016. So this program actually really benefited me a lot in different ways. So that was my primary funding.
Yeah, great. So this interdisciplinary approach, I think, is always great and really teaches you a lot also how to think about your project, I think. So very nice. So maybe looking ahead a bit. So how might your research be applied in a clinical setting, for example? So are there any potential real-world medical benefits, or do you think it's possible to maybe selectively target CFH in the hematopoietic compartment, or will it be possible in the future?
Yeah, this is, of course, a very important question. And in general, complement therapeutics have gained a lot of interest in the past, let's say, decades. And there are, FDA-approved drugs out there that target complement at different levels. Also, there's a new one which targets complement at the level of C3. So something that can be interesting in this regard. These are, particularly effective in autoimmune diseases where complement attacks the host.
Sure.
And however, when it comes to our findings, for example, I find this a little bit tricky because, our data clearly show that monocyte-derived CFH has a pro-inflammatory effect. But we also see, and this is also, in the paper, that liver-derived systemic complement factor H has an atheroprotective effect by limiting systemic complement activation. So, the challenge is still there that one would need to target CFH in a context and also a tissue and even cell-specific manner. So that can be challenging. But for example, in a recent publication that I just saw it, and it seemed to be partially motivated by, some of our findings, a group used, for example, siRNA-loaded liposomes, and they silenced local CFH production in a pulmonary fibrosis model. So that, that's an option. Or they saw, favorable results. And obviously similar immunotherapeutic approaches, such as lipoprotein-based nanoparticles, are available in atherosclerosis research as well. So and then important to mention that also these nanoparticles, they preferentially target monocytes and macrophages. So either local CFH silencing or promoting local C3 activation in a way, basically using these approaches could be feasible. However, obviously we are kind of far from that at the moment.
Yeah, but maybe one day. There's hope. No, that would be fantastic, of course. So what is next for this project? I mean, you already mentioned many options, but do you already have a specific follow-up study planned, or have any recent publications beyond the one you've mentioned in lung fibrosis? So have any other recent publications maybe expanded on your findings?
Oh, yes. Not from our lab yet, but we are working on that. But luckily, I have to say that we see that it's, the study is fairly well cited, and there are, labs that kind of base some of their approaches, maybe the idea, that cell autonomous complement regulation can occur. And obviously, we are also constantly thinking about potential follow-up studies. So one direction that for us could be particularly interesting to link malondialdehyde, so MDA binding by CFH, to the cell autonomous effect that we see on complement. So, because this, I think, could have implications in atherosclerosis. You could think of like how local availability of macrophage-derived complement can be dependent on the extent of apoptotic cells or local necrosis, because these apoptotic cells, for example, would be decorated by malondialdehyde and other oxidation-specific epitopes. But we still need to test this. Another topic that I have been exciting about is the role of cell autonomous complement in the central nervous system, especially in brain border regions, which are important neuroimmunological niches. Because, we know obviously a lot already how important complement is in the central nervous system. You can think of neurodevelopment, you can think of synaptic pruning, or clearance of danger-associated molecular patterns. Importantly, also, there have been evidence in an hetero setting. If you think of, for example, recent work by Andreas Habenick's lab, who showed that complement dysregulation can occur at the choroid plexus, and that can contribute to beta amyloid accumulation and also atherosclerosis. So, however, what I see, or when I, have a glance at the literature, that no one seemed to look at complement production by macrophages or monocytes at the brain borders, which would be, monocytes or even border-associated macrophages in the brain or in the skull bone marrow or in the meninges. And so we have collected some preliminary evidence that C3 and CFH are produced at these sites by macrophages. And the next step would be to find out how they are involved in, immunological responses at the brain border, or whether they are implicated in neurodegenerative diseases, or even in cardiovascular diseases like stroke.
Yeah, that's very exciting. And it seems you really embrace this interdisciplinary research approach that you've already talked about as part of your PhD program. So it sounds like you've become very interested also in the neuroscience field.
Exactly. That I have to thank also to Phil.
Yeah.
He's also a big supporter of the interdisciplinary thinking.
Oh, that's great. So maybe related to that, looking ahead, what are your long-term research goals, and what kind of impact do you hope that your work will have over the next decade or two?
Yeah, so currently I can tell you, but you know that my main focus is grant writing. So I would like to establish myself as an independent investigator, start my own lab. And this is, where I would like to combine my expertise and interest, which would be, combining atherosclerosis research with immunology or neuroimmunology and neuroscience. And as I mentioned before, I'm particularly interested in the key neuroimmunological interfaces in brain border regions. And I would like to understand how they could be implicated in both central as well as peripheral atherosclerosis. And for my future, I only hope to do exciting science and that which kind of would involve this immunological axis between the brain and the heart. So kind of brain and heart communication. And also eventually, obviously, I would like to surround myself. I'm already surrounding myself, but actually, by establishing my own lab, I would like to find some enthusiastic people who are interested in this angle. And my main wish would be to be able to, motivate and mentor young scientists and contribute by this to the scientific community.
That's really great to hear, because I'm sure I'm not just speaking for myself when I say that many young researchers really look up to young scientists like you. So is there any specific advice you would maybe give to someone who's really just at the beginning of their research career?
Yes, I usually don't like to give advices. I'm asked, but as you asked me, I can think of a couple, obviously. First and foremost, I think it's very important and that don't be afraid to ask questions. This is obviously a bit, if I think of myself, is actually a bit hypocritical, because I've been always struggled with this and still kind of struggle to ask questions sometimes when I don't feel like it's completely backed up by some recent evidence in the literature. But I think it's very important, because you have to make your ideas heard, and even, in a less mature thought that you have can fuel a great discussion and culminate in something new. Another advice that especially during the PhD, that don't isolate yourself, I would say. So oftentimes I think, especially during the PhD, one might think this is never going to end, or you face its challenges, obviously, maybe reminiscent of some that I faced before, but I really had a good support. And then, you might think this is up for me to resolve these issues. However, I think it's always worth asking for help. And again, wherever I went so far, I was always very happy to realize how many people are there who are open and selfless, and they're ready to give you a helping hand if you need. So I think that would be it.
Yeah, that's great. And of course, always very important to surround yourself by the right people, and also realize that you have a great network around you. Exactly. So that's really great advice. Thank you. Would you say you've already found your research niche? And if so, how did you really define it as your focus? Because this is, of course, also something that particularly young researchers are always asking themselves.
Yeah. So currently, I believe so. I feel quite happy in my research niche at the moment, which is basically a blend of my predoctoral and postdoctoral work. So on one hand, I think it's kind of safe. I have the safe grounds of immunology and atherosclerosis research, where I feel confident, in my daily work and operations. But on the other hand, I have tremendous amount to learn regarding neuroscience. This is something that I've never been properly, taught. And then this kind of, when you look at the heart-brain axis, this is definitely something that I'm trying to, improve myself, which makes kind of every day exciting and is kind of a constant intellectual challenge.
Yeah, that's great to hear. So, yeah, maybe just one more question on this. So, one fascinating aspect of your work in the past has also been your involvement in research on sleep and atherosclerosis. So I know you've done a lot of this work with Cameron McAlpine in Phil Swierski's lab. Could you maybe also share some of the interesting observations you've made in this field?
Yes, I'm happy to. So, as you said, I got involved in the sleep research during my semester abroad. And so we contacted Phil and he said, I have a new postdoc here. He just did an experiment where he looked at sleep, and what he found that sleep fragmentation results in increased atherosclerosis, like a kind of a groundbreaking finding that, has
great for PhD students.
Had never been shown before. Yeah, exactly. And that was obviously Cameron McAlpine who really spearheaded, the sleep research in Phil's lab. And so that was the initial finding, and eventually working together, we kind of worked out some of the mechanistic insights of this study. So what actually Cameron found that in 16 weeks of sleep fragmentation in mice leads to decreased production of a neuropeptide called hypocretin or orexin in the lateral hypothalamus. And interestingly, this hypocretin signals to the bone marrow, and what it does, it basically modulates hematopoiesis. So in the setting that we had in heterogenic mice, sleep fragmentation, you had, due to the decreased availability of hypocretin, you had increased hematopoiesis, so proliferation of hematopoietic stem cells, and that led to increased number of monocytes and neutrophils in the circulation, and eventually increased infiltration into atherosclerotic lesions, and as a consequence, increased lesion size. And this was also the case later on we did in hypocretin deficient animals on an atherosclerosis prone background. So that was the first study, and then it was already during my postdoc stay when we had a follow-up study where we could also answer a couple of additional interesting questions. First, what Cameron already did is basically he gave the mice the opportunity for recovery sleep. And then interestingly, it takes 2 or 3 weeks for the sleep quality, to actually come back to the baseline. And even after 4 weeks of sleep recovery, the number and the proliferation of these hematopoietic stem cells in the bone marrow, they still remained elevated. So these findings really indicated to us that sleep has like a permanent impact on hematopoiesis. So, and indeed what we found later on, that sleep fragmentation exerts really like a long-lasting effect, and this is by restructuring the epigenome of these hematopoietic stem cells. And accordingly, so when you subject these mice to a secondary challenge after sleep fragmentation and recovery, this actually results in exaggerated inflammatory burst and worst outcomes, which then later on we could mitigate with epigenetic modifiers. So that was the second study. And then when I basically moved to Phil's lab, the aspect of my research was to also understand what effect permanent sleep fragmentation has on adaptive immunity, because this is something that we didn't really look at before, just partially. And so we basically did a huge immunoprofiling, and then what we found that sleep fragmentation actually specifically affects or leads to maladaptive B-cell responses in brain-draining cervical lymph nodes, as well as in the meninges or the dura of the meninges, which is a critical site of neuroimmunity, and through the meningeal lymphatics also drains macromolecules and waste products to the brain-draining cervical lymph nodes. So this is currently the project that we are trying to finish, and we have already looked, in this context to neuroinflammation, and we have evidence that sleep fragmentation instigates, for example, microglia reactivity, perturbs lymphatic flow in the central nervous system. And we believe that these maladaptive responses that we see in the meninges as well as in the cervical lymph nodes are downstream effects to basically what happens in the brain. But these findings are yet to be published, and hopefully we can finalize the story.
That sounds very exciting. So good and consistent sleep hygiene is really important.
Yes, definitely.
And it sounds like a big topic that I hope we will maybe cover in one of the future episodes of this podcast, a bit more extensively even, but very exciting work. So, okay, so as we wrap up, is there a particular paper or even book that has been especially influential in your work, or something you'd recommend to our listeners?
So I have to say, when I used to be a teenager, I really read a lot, but in general literature. Yeah. And unfortunately that, has dropped ever since. So the recent books that I've read, they mostly have to do with my research, which is, I don't know if it's good, but especially in particular sleep. So there's a book that has been sitting on my bench in the past years. So this is called Sleep. Mm-hmm. And this is by J. Allen Hobson. And this is a sweet book to me because my girlfriend and I picked it up in an old bookstore during one of our long walks, actually, in downtown Boston. So this was close to this Old South meeting house where there is this old bookstore. We found a copy. Interesting, it was published in 1989, so even before I was born. So it's very interesting, to see how much there had been already known even before I was born about sleep. But it's also kind of remarkable how much ground we have covered since. So this is an interesting read. And for a light read, I think my recent book that I read is Matthew Walker's book which is Why We Sleep, which is a popular science book, that was written specifically to the general audience. And I mean, it's, obviously it's written for the community. So it's not necessarily, perfect, always, if scientifically speaking, but I really enjoyed reading it because it really communicates the importance of sleep. And I actually do a lot of car sharing when I travel from one place to another. And then you meet a lot of people that of different, people with different backgrounds and professions. And they often ask about what do you do, and I say, well, I'm a researcher. What do you research? I mostly sleep. And then this can be really like a conversation trigger. Sure. And that's funny. And most of them, what is, I think it's always nice to realize that most of these people seem genuinely open to hear about science, and they can ask actually very straightforward questions. And for example, this book that I just mentioned, which is again called Why We Sleep, it really helped me because, it helps you to channel the general concepts and gives you ideas how to communicate to a non-scientific audience. But which is important because, as you just said, I mean, we need to consider the importance of sleep, not only in the general audience, but also I think in a scientific setting, because to date, I attend sometimes meetings or, there are these meet the speaker sessions after a talk. And then there's still examples when an individual would basically kind of brag about the fact that I only sleep 5 hours, or I start writing emails at 3 a.m. And I think this is a pity because, at this point, I think we have enough scientific evidence which underscores the beneficial impact of sleep on our health, and especially, I think, in cardiovascular research. And basically, unless, you are a natural short sleeper, which can happen, but it's only like,
one exception probably.
Yes, it's like 1% to 2% of the general population, I believe. So then if you are not in this 1 or 2%, 5 hours of sleep is probably not enough for you to maintain a good cardiovascular fitness. And what if I could mention, actually, this was the latest that I finished. And then, so the most recent book that I have in my hand, which I very, very recently started reading is The Principles of Neuroscience. This is written by Eric Kandel, and I received a copy of it from my colleague in New York, Matteo, who is a reading machine, and he's my dealer. He gives me books, he gives me papers, and then I try to keep up with him. So right now, as I'm trying to deepen my understanding in like hardcore neuroscience, I'm particularly excited about this book, which was again written by Eric Kandel, which obviously then also for us it has...
So now in Vienna.
Exactly. So it has a sentimental element to this as well, because as many of us know, like Eric Kandel is a Vienna-born Nobel laureate, and then that makes us, feel also the book closer to us. And actually, in his honor, we also have a new institute opening soon at our campus, which was going to be called Eric Kandel Institute for Precision Medicine. So it's exciting times, and hopefully this soon opens, and we are looking forward to having enthusiastic scientists joining us from all around the world here in Vienna.
For sure. I know who I will be coming to for book advice in the future. Okay, so finally, we always have one short part of this podcast dedicated to failures, which we call the graveyard of ideas. So most of the time, we only hear success stories of other researchers, which in a way makes some of us also question ourselves sometimes. And, I think it's really important for all researchers to know that really failure is part of the process and you really grow from it, right? So, that's why we decided to ask everyone in this series of interviews to also contribute an idea to our graveyard of ideas. So this could be a story about, for example, a grant you were passionate about that maybe in the end didn't get funding so far, or any kind of failure that you faced during your academic journey. So is there anything you can contribute to our graveyard of ideas?
Yeah. I guess definitely, because as I mentioned, maybe I can channel this back to the paper that we just talked about recently. So obviously, having an unexpected observation and finding a new hypothesis always comes with some ideas that you try and maybe don't, hold true. So this would be, for example, with the CFH, we had many of these. Like we thought at one point it may have like a chemotactic function in like instructing monocytes to infiltrate the lesions. Then at one point we looked at like monocyte B-cell interaction. That was not the case either. So definitely behind this story, even there is like a graveyard of ideas. And then it's quite a big graveyard, and so it took eventually, quite some time for us to actually conclude this project, which was almost like I started my PhD in September 2013, and then officially the paper was published in August 2023, so almost as a 10-year anniversary special. But also you could mention like grant submissions. Obviously, when I moved to the US, I tried the postdoctoral fellowship for the American Heart Association, and I received quite good scores at the first try. So I was really confident, like this sounds good. There were some, minor suggestions. I addressed them, submitted the next year, didn't even get discussed, so this can happen. And and I think failure, and also it's kind of, like a failure can really feel like a failure at the time, but maybe, it's the beginning of something new. And then not every success story starts with a success story. That was definitely the case for me. Maybe for this audience, it's important to mention that at the beginning of my PhD, I feel like I struggled for a while. I was not necessarily up to the challenge, to the point when, this was addressed. We discussed this with Christoph, we discussed my, future in science, which was eye-opening. And then, I just realized that how I can, put more effort into kind of coming across as a dedicated and determined person. And then we met regularly. We discussed in details the experiments that we did. We did this for a while, and it really paid off. This was really helpful. And then eventually, look at us, here we are again, working together still after over a decade. So even though, sometimes might feel you are down or things don't go your way, eventually there's going to be, if you put, I think, the right effort, then there can be like a really, it can be a game changer, and it can end up in your own success story, hopefully.
Yeah, that's very encouraging to hear. Thank you so much. So yeah, Dr. Kish, Mate, thank you again for joining us today. It's been a real pleasure speaking with you. And yeah, also to our listeners, thank you so much for tuning in. And please don't forget to subscribe for more insightful conversations with other leading researchers in the field. So until next time, take care and thank you, everyone.